Hi everyone, welcome back. We're starting a new cycle. First, we started out with glucose and how we get energy, catabolism of glucose, and we go through 10 different steps so that we can get through to a 3-carbon molecule of pyruvate. And after we create that 3-carbon molecule of pyruvate, we can continue under anaerobic conditions and make lactate, that's fermentation, or we can take it into the citric acid cycle. So the citric acid cycle is under aerobic conditions where we take that 3-carbon molecule of pyruvate and get more energy out of it. So we'll end up getting to the very first steps of the citric acid cycle. But for now, we're going to go do a little glycolysis review to kind of orient us where we started and where we want to end up. Hopefully by now you have memorized glycolysis, but just to remind you, there are two phases of glycolysis. There's the preparatory phase when we have to invest 2 molecules of ATP, and then there's the payoff phase where we gain 4 molecules of ATP and 2 molecules of NADH. So as I like to do, I like to memorize the enzymes, and that helps me to remember what are the reactions that are taking place. So I know we start out with glucose. You'll be given a whole page, a cornucopia of different structures, and you should be able to recognize where they fall in these different cycles and phases, and the enzymes that catalyze the reactions and the products and reactants. So anything could be missing: the structure of the substrate, the structure of the product, the enzyme, and you should be able to pick out and choose from a very long list of structures so that you can place them in the cycle. But anyway, I know that the first enzyme is hexokinase. Good. Then so it's a kinase. So I know that it's going to take a phosphate from ATP and add it onto my substrate. So I'm going to get G6P. So here we are, G6P. I know the second enzyme is an isomerase, phosphohexose isomerase. So we're going to create F6P from G6P. I know that the third enzyme is PFK1. So PFK1 is a kinase as well. So we're going to phosphorylate IF6P and create F16BP. And so I just described the first step of glycolysis and the third step of glycolysis, but rather than going over gluconeogenesis, I want to just point out that the first step in glycolysis is irreversible, and so it's a last step in gluconeogenesis. Since it's an irreversible step, it's a regulated step, and we cannot use hexokinase. We have to utilize a different enzyme in gluconeogenesis, and that enzyme is glucose 6-phosphatase. So we're going to go in gluconeogenesis from G6P to glucose. PFK1, you should make sure you remember that it's another regulated step, and it's irreversible in glycolysis, so we're going to utilize FBPase1 and gluconeogenesis. Okay, so now we've created our doubly phosphorylated F16BP. So we should recognize the structure here, F16BP. And once we create that, then we're going to chop it in half. We're going to utilize aldolase. So aldolase is responsible for creating 2 3-carbon molecules, both phosphorylated. You don't need to know the name of these. GAP and DHAP are good enough names. 2 phosphorylated 3-carbon molecules. And then the last enzyme is triose phosphate isomerase. Yes. And that is going to give me go from DHAP to GAP, so I have 2 identical molecules of GAP created from the 6-carbon molecule glucose. Now in the second stage, we call it the payoff stage, and so we are going to actually generate ATP. We're going to generate 4 molecules of ATP and 2 molecules of NADH. So if we start with a molecule of GAP, the very first phase is going to give us our NADH. So if we utilize NADH or NAD+, Then we're going to utilize the dehydrogenase, our GAP dehydrogenase. You can just memorize it as that. And then the next enzyme is phosphoglycerate kinase. And so now we know it's a kinase, but it's named for the reverse reaction. And I know that because we're in a payoff phase, so we're going to create ATP. So we're going to take in an ADP. We're going to take off the one of the phosphates from the phosphoglycerate and give us 3-glycerate. The next step utilizes phosphoglycerate mutates. So we're going to go from 3-phosphoglycerate to 2-phosphoglycerate. You should remember for this step that we are utilizing this mutase. The reaction mechanism is important here because we have a phosphate on the histidine, and that histidine is going to put a phosphate on the second carbon of our 3-phosphoglycerate, and we're going to create 2,3-BPG. So if we remember that 2,3-BPG allosterically modulates hemoglobin, and so if you're creating a lot of 2,3-BPG, you're going through glycolysis, you need a lot of oxygen, you need a lot of energy, and that triggers hemoglobin to release oxygen. So that mechanism is important. And then the next step utilizes enolase. We should remember in that step we're going to release water. And then the last step in our payoff phase utilizes pyruvate kinase. And again, this one is not only named for the reverse reaction because we know we're getting ATP, so it's the reverse reaction, but also it's a regulated step in glycolysis. And because it's an irreversible step in glycolysis, we can't use the same enzyme in gluconeogenesis. We actually are utilizing 2 different enzymes, and those enzymes are PEP carboxylase and pyruvate carboxylase. I couldn't remember those 2. So in gluconeogenesis, since this is an irreversible step, we utilize a different enzyme. We don't go from pyruvate to PEP. Using pyruvate kinase, we have to use 2 decarboxylases in gluconeogenesis. So all those things you should remember. You should also remember, as a review, another objective we had was to learn the feeder pathways into glycolysis. And so we learned glycolysis is 10 steps. We get pyruvate, and utilizing glucose. But there are other carbohydrates we know that we eat. We eat a lot of glycogen. We eat a lot of starch, actually, from potatoes and wheat and rice and all that stuff. We may drink a lot of milk or dairy products where we get lactose. We might eat a lot of sugar where we would end up with sucrose. And so all of those carbohydrates end up going into the glycolysis pathway, either by being converted into glucose or be converted into an intermediate in the glycolysis pathway. And so you should be familiar with certain things about our intermediates in the glycolysis pathway. So the structures in pink you should know already, so you should be able to recognize those. The structure of fructose, the structure of sucrose, the structure of glucose. You don't need to know the structure of trehalose. It's a disaccharide that you weren't expected to learn. The structure of lactose, the structure of galactose, and you weren't asked to learn the structure of mannose, but it's just a epimer of glucose. But you should be able to recognize and be familiar with these structures, and also how do they get into glycolysis. So I'll just review that very quickly. I'll start with this one. Trehalose, it's a sugar that you would find in trees. And very simply, how do you get into the glycolysis pathway? Well, trehalose gets broken down by trehalase, and then we get glucose. Straightforward, simple. What about lactose? You know what lactose is? It's the sugar that's in milk. So it's a disaccharide. So first we've got to break that disaccharide bond. And it is a disaccharide of glucose and galactose. Glucose is great. It's the first step in glycolysis. But then we also have galactose. So what you should remember about galactose is that through a series of steps, it gets converted into G1P, glucose 1-phosphate, and then through one more step, it goes into G6P. So skip all of this stuff. First we go to G1P and then G6P, which you know is the second step in glycolysis. It's the substrate in the second step in glycolysis. Alright, so we also utilize sucrose probably. And so sucrose, since it's disaccharide, we have to break that glycosidic bond. We use sucrase. And what is sucrose? It's a disaccharide of glucose in glycolysis. Glucose, of course, is fine, but fructose has to enter the glycolysis pathway. And so we can use hexokinase. It's a ubiquitous enzyme. It phosphorylates glucose and it phosphorylates fructose. And when we phosphorylate it, we add a phosphate to the 6 carbon again, and we get F6P. And of course, that's the third step of glycolysis. Alright, so we talked about trehalose, sucrose, lactose, galactose. And lastly, there is glycogen and starch. So glycogen and starch are monosaccharides. We usually store glycogen, but we could eat some too if we're eating another mammal that has glycogen stores. But starch is just a polysaccharide of glucose. So one of those is amylose. So amylase breaks that down into glucose molecules, and it goes right into the citric acid cycle. And then the last one, mannose, which is in our diet some kind of way. Mannose gets phosphorylated by hexokinase again. We get M6P, and M6P goes right into F6P. So mannose becomes M6P, and M6P becomes F6P. So make sure you are familiar with all those feeder pathways into glycolysis. Also, make sure that you review this slide, in particular, the regulation of glycolysis as well as gluconeogenesis. And since glycolysis is for energy production, it gets activated when we need energy, such as when there's low ATP. We'll talk about acetyl-CoA as well today. Acetyl-CoA is the first molecule that goes into the citric acid cycle, which gives us more energy. So if we have low storage of acetyl-CoA or low citrate, which is where the name citric acid cycle comes from, we need to do more glycolysis, and it's inhibited by the opposite. Otherwise, when we need gluconeogenesis to maintain our glucose homeostasis, we don't need too much glucose in our bloodstream. Maybe we need to turn that into glycogen. Then it's regulated by the things that are, or inhibited by the things that are activated by glycolysis. So make sure you're familiar with this slide and can answer questions related to it. Remember that after we've created pyruvate, we can actually go through fermentation or cellular respiration. So it's called fermentation in the absence of oxygen. The whole process of glycolysis all the way up to the formation of lactate is sometimes called fermentation. It means under anaerobic conditions without oxygen. So we should remember the structure of pyruvate, and that when we are sprinting or there's just not enough oxygen around, we can convert that pyruvate to lactate, utilizing lactate dehydrogenase. And so I know when I see dehydrogenase, I'm going to utilize an NADH or NAD plus or something like that. So we're going to take NADH and create NAD plus in the creation of lactate. Yeast, on the other hand, would take pyruvate under those anaerobic conditions and first decarboxylate it with pyruvate decarboxylase into acetaldehyde, and then into ethanol utilizing dehydrogenase. And so we do this for a couple reasons. One is because there's not enough oxygen around in order to go into the citric acid cycle, but we also want to regenerate our NAD+. So we see that in mammals and also in yeast and some other microorganisms. We need to regenerate that NAD+. because we can utilize that in glycolysis again if we need to break down more glucose, or if we need to break down more glucose and get energy under those conditions. And so we usually go through pyruvate and lactate, but remember that we also would have this reverse reaction from ethanol to pyruvate. If we drink alcohol, then we would need to utilize an alcohol dehydrogenase in order to metabolize that into pyruvate. Now we reviewed glycolysis. We know we start at a 6-carbon molecule and we end up with 2 3-carbon molecules of pyruvate. And the purpose of going into the citric acid cycle is so that we can get more energy out of that glucose molecule. And so our learning objectives will be broken up in 2 parts or 2 lectures. First, we'll be reviewing section 16.1. It's the production of acetyl-CoA. And so it's a very complex process how we actually even get into the citric acid cycle from pyruvate. So it must be converted first into acetyl-CoA. So we'll talk a lot about the enzymes, the coenzymes, the cofactors, and the sequence of events that allow us to get from pyruvate to convert it into acetyl-CoA that can then be condensed into molecules to enter the citric acid cycle. And then in the second lecture that will be coming up next week, we'll go over the reactions, the substrates, the products, the enzymes, just like in glycolysis, that are in the citric acid cycle, and then briefly talk about regulation of the citric acid cycle. Now, before we go into the citric acid cycle, we just want to be reminded that pyruvate is a branching point. So pyruvate can be a source of glucose from gluconeogenesis. We have enough energy. We can store that glucose as glycogen so our brain or whatever needs to use it later. Also remember, another objective was to learn the pentose phosphate pathway. Very little about it, except that it generates NADPH and it also helps to generate ribose. So ribose is the sugar in our nucleotides, our nucleic acids in our DNA and our RNA. And so we can generate ribose from glucose. Or, and this is what we'll talk about today, or pyruvate can be a source of acetyl-CoA. And so you take the pyruvate, decarboxylate it. So we see that here. Pyruvate at the end of glycolysis can be decarboxylated and attached onto a coenzyme A. So we create acetyl-CoA. And then we get a lot of energy by going into the citric acid cycle. Now, if we don't need energy, we can create acetyl-CoA. And we know that acetyl group is just a 2-carbon compound. And that 2-carbon compound can be stuck with a whole bunch of other 2-carbon compounds and make fat or lipids. So we want to store our energy. Our long-term energy source is our fat. So we can take the pyruvate, decarboxylate it, make acetyl-CoA, and that acetyl-CoA can help us to make fat for storage. Or it can make ATP in the citric acid cycle. Therefore, we'll see a lot later on that acetyl-CoA works to regulate these options, whether we want to store fat, whether we want to store glycogen, glucose as glycogen, or whether we need actually more energy. So if our body is undergoing cellular respiration rather than fermentation, that's the process by which our cells consume oxygen, produce CO2. We need energy, and that's the preferable source of our glycolysis, burning that energy from glucose in our citric acid cycle. It occurs in 3 major stages. So one of the stages, this picture has a lot going on, but it's very simple. One of the stages we talked about is glycolysis, 10 steps, pyruvate. And then we can only enter the citric acid cycle by converting pyruvate into acetyl-CoA. We get a lot more energy. So the first step is glycolysis. Our fatty acids do not undergo glycolysis. The fatty acid part can be converted 2 carbons at a time into acetyl-CoA, and the glycerol can go into glycolysis through some steps. Our amino acids can be converted into acetyl-CoA Many times our amino acids can be converted to many of the intermediates in the citric acid cycle, but they also don't go through glycolysis. So the acetyl-CoA is kind of a bridge point where all our macromolecules that we eat, our sources of energy, carbs, fats, and proteins, get converted into acetyl-CoA before they can get energy and go through the citric acid cycle. So that is a very important step. So the acetyl-CoA enters the citric acid cycle. A lot of what we get from the citric acid cycle, some of it is energy, but some of it is electrons in terms of our electron carriers, or NADH or FADH2. And those can give us more energy in oxidative phosphorylation, that's stage 3. So most of our energy, or our ATPs, are going to come out of oxidative phosphorylation, which comes up in about 3 chapters. But now we're at the point where we have to enter the citric acid cycle, cellular respiration, where oxygen is abundant, so we can get more energy out of glucose. So as I said, our first and only objective we're going to cover today is the conversion of our 3-carbon pyruvate, which we know the structure of, into acetyl-CoA. And so acetyl-CoA gets us into the citric acid cycle. It's just the in-between where we can get our pyruvate that we created in glycolysis into the citric acid cycle. Some points to notice about that is the pyruvate, this conversion happens in the mitochondria and it takes place utilizing a very large complex of enzymes, so many enzymes that are working together in order to convert this pyruvate to acetyl-CoA. It's a completely irreversible reaction. It has a very large negative delta G double prime knot. And the pyruvate dehydrogenase complex, we'll go over a lot in a minute. It's 3 different enzymes, E1, E2, and E3. And the net reaction is an oxidative decarboxylation of pyruvate. So we see the decarboxylation of pyruvate, so we lose the CO2. And either we can take this acetyl-CoA and enter the citric acid cycle so we can get energy, or we can take this acetyl-CoA, if we don't need energy, and turn it into a lipid, into fats. And so this complex utilizes these 3 enzymes, but it also utilizes 5 coenzymes or 5 cofactors. And we'll learn about each one of those individually. So the first cofactor is the actual coenzyme A. So sometimes you see CoAsh or CoAsh, just to emphasize that the SH is involved. But this is coenzyme A. It utilizes a NAD+, the second coenzyme. It utilizes a TPP, a lipoate, and an FAD. So it doesn't seem like a lot is going on. You're just going from pyruvate to acetyl-CoA, but it's a very complex set of reactions utilizing 3 different enzymes and 5 different coenzymes. And so we'll go over each of those individually. How do we get from pyruvate to acetyl-CoA? We utilize a pyruvate dehydrogenase complex. It's a complex of 3 different enzymes with many different complex copies. Many different copies of 3 different enzymes in this complex. And it's so large, about 7,800 kilodaltons, that we can see it easily on an electron microscope. So this is pretty fascinating to be able to see a protein that's so large we can see it on an electron microscope. So here's just a cartoon picture of this complex, the 3 different enzymes, E1, E2, and E3, and it's just copied, tens or hundreds of times depending on what organism it is. So what you should know, there are 3 enzymes. We just call them E1, E2, and E3, and each of these contains some of the cofactors or some of the coenzymes. So E1 contains the TPP cofactor. We'll go over that. E2 will have steps 2 and 3, and we'll have lipolyl cofactor, and we'll have the coenzyme A, and then enzyme 3 will have the FAD cofactor and the NAD plus co-substrate. So again, this is just to point out how massive this complex is, and that not only is it massive on its own, it also employs 5 different coenzymes, and that's TPP, lipoA, coenzyme A, FAD, and NAD+. Let's go over the sequence of events for the conversion of pyruvate to acetyl-CoA. So I'll go over these quickly at first on this slide, and then each step individually. So don't get worried if you get lost on the first slide. Let's just go through an overview of all the sequence of events and the steps, and then we'll go over each step individually. Okay, so first we know that we have our pyruvate. And in step 1, we're going to be in the E1 enzyme. So this is E1. And E1 has the coenzyme TPP. So we see TPP is in E1. So what happens is first we have decarboxylation of pyruvate, so we lose the CO2. And then we form the hydroxyethyl TPP. So basically the CO2 leaves and the acetyl group attaches to, or the hydroxyethyl group attaches to TPP in the E1 enzyme. So that's step 1, decarboxylation and the formation of the hydroxyethyl, the 2-carbon part of pyruvate, attaches to TPP. Step 2 is a series of events. We have oxidation of the hydroxyethyl group. Then we have reduction of the lipolysine. So we'll see a structure of that. It's just basically a lipolysine, lysine with some other modification to it, and it's going to attach to that lipolyl lysine, that modified lysine. So we're going to form our acyl lipolysine. So step 2, we're going to move this 2-carbon part of pyruvate from TPP to acyl lipolysine, and that's going to be in enzyme 2, E2. And then we're going to go straight into the formation of acetyl-CoA. So by step 3, we formed acetyl-CoA. So we transferred from an acyl lipolysine to acetyl-CoA. Right now, we formed our acetyl-CoA, but we need to regenerate our enzyme because we have we reduced the lipolyl lysine, so we need to oxidize it back to where it was. And we're going to use our cofactor FAD. So now we need to regenerate that lipolysine. We're going to utilize FAD and create FADH2. So reoxidation of the lipolysine, that's in the E3 with FAD. Now we created FADH2, and that's wrong, so we need to regenerate our FAD. And we're going to regenerate that using NAD+. So now we've gone back to FAD in step 5. We've gone from NAD+ to NADH. And NADH is something that we can utilize later as an electron carrier. So everything is regenerated. Acetyl-CoA is created. And we've utilized all of our 5 different cofactors, ending with NADH, which can be an electron carrier that we'll use in oxidative phosphorylation. Let's go into detail of step 1. So step 1, we started with the pyruvate. We decarboxylated the pyruvate, and then we made this thing right here, this hydroxyethyl thiamine pyrophosphate, TPP, hydroxyethyl TPP. So step 1 was decarboxylation of pyruvate, loss of CO2, and creation of hydroxyethyl TPP. So TPP, which you don't have to know the full name, but you should know TPP, is a coenzyme form of vitamin B1. So vitamin B1 is found in food, of course. It's found in whole grain, rice, and wheat and stuff like that. And so when you have white rice and polished rice, which most of the world eats a lot of, they can be vitamin B deficient, and then they can't go through this decarboxylation part of the pyruvate dehydrogenase, and they can have some neuronal deficits and diseases from that. So it's really important for people to get this vitamin B in their diet so they can have TPP, so they can perform this function. All right, so what happens mechanistically is this TPP has this thiazolium ring. So the thiazolium ring, it loses its hydrogen. So when it loses the hydrogen, now we have an anion. So we have this carb anion. And then this carb anion attacks the carbonyl carbon. This is kind of crazy here. The carbonyl carbon of the pyruvate. That looks like any word at all. All right, so we have this carbon anion that attacks the carbonyl carbon of pyruvate, and then we have this hydroxyethyl thiamine pyrophosphate, this hydroxyethyl TPP. So basically, and then the CO2 leaves, so the CO2 is released. So we created this hydroxyethyl TPP in the very first step of or the very first sequence in events of trying to get from pyruvate to acetyl-CoA. In step 1, we had hydroxyethyl TPP, a decarboxylated form of pyruvate, is stuck to TPP in the E1 enzyme. Step 2 happens in the E2 enzyme of the pyruvate complex. So eventually we're going to get to acyl lipolysine, but let's see how we get there. So we know what lysine is. It's an amino acid that's sticking out of the enzyme in the E2 enzyme. But it's modified to have this lipoic acid. So we have this lipoic acid or this lipolysine. All right, so the first thing that happens is we reduce the lipolysine. So we have the reduced form of lipolysine. That makes it so the aldehyde can be oxidized, and it carries the acetyl group for transfer. So we have an acetyl group from the hydroxyethyl TPP. It gets transferred to the lipolysine, and now we've created the end product of step 2, the acyl lipolysine. So we're just kind of transferring things along. We have the pyruvate or the decarboxylated pyruvate on the hydroxyethyl TPP. Then we moved it to E2, and we're at an acyl lipolysine. Now in step 3, we have the acyl lipolysine in E2, and in step 3 we go straight to the transferring the acetyl group to acetyl-CoA. So now by step 3 we created acetyl coenzyme A. This is the structure of acetyl coenzyme A. It's involved, the thiol group is involved in the transfer of the acetyl group to the coenzyme A. It doesn't really matter what the structure is. It's just very complex. It's a coenzyme, and it gets stuck onto the acetyl group, and that's where we what we do with that pyruvate. So our coenzymes or our co-substrates are not permanent part of the enzyme structure. They can associate, fulfill a function, and dissociate. So we see that this one is going to dissociate. It can be regenerated and come back into the enzyme and do other things. But the function of coenzyme A is to accept and carry acetyl groups, and that's what it's done. It's accepted acetyl group and it's carrying it along into the citric acid cycle. Now that we have utilized or created our acetyl-CoA, then we need to regenerate our enzyme. And so step 4 and 5 are into regenerating our cofactors. Step 4 is to the reoxidation of the lipolysine. So we reduced it before, and we need to get it back to its original form and reoxidize the lipolysine cofactor. And we're going to utilize a FAD. You might have heard of FAD. We'll see that a lot later in oxidative phosphorylation in the citric acid cycle. And FAD actually has two different nucleotides. The first nucleotide is FMN, and it has a nucleotide called riboflavin. So we know that a nucleotide has a sugar and a base and a phosphate. This is called riboflavin, sorry. The sugar is a ribitol, a reduced form of ribose. We see the sugar here. We see the base is 3-fused-ring. This is called flavin. And we have the phosphate group. So FAD contains an FMN, as well as, hopefully you recognize this nucleotide. The second nucleotide is adenosine monophosphate. So we have our adenine, our ribose, our phosphate, and an AMP. So if you stick an FMN and an AMP together, you get FAD. And FAD acts as an electron carrier so that we can perform oxidation and reduction reactions. And so we're going to have our FAD being fully reduced to FADH2. The same would happen if you just had the FMN molecule. Well, we're going to utilize FADH2 and fully reoxidize our lipolysine. So that's step 4. So if you know that we have to regenerated our lipolysine, but now we have to regenerate our FAD. And so we do that in step 5, right? Regeneration of the oxidized FAD cofactor. We created a FADH. We need to regenerate FAD, and we're going to utilize an NAD+. So the same kind of reactions apply. So I won't go through any of that, but just to say we regenerated our FAD by taking an NAD+ and creating an NADH. And so in the end, the only thing that's not regenerated, or not the same as in the beginning, is that we created an NADH from NAD+, and that NADH is going to be an electron carrier that we can put into our oxidative phosphorylation pathway and get energy out of that later. So again, we should make sure we understand each of the steps in the sequence of going from pyruvate to acetyl-CoA. First, we decarboxylate. We get TPP, or hydroxyethyl TPP. That's an E1. In E2, we end up with the acyl lipolysine. And in E2, we transfer that acyl group and make acetyl-CoA. Then we need to regenerate our lipolysine with FAD, and then we need to regenerate our FAD with NAD+. It looks harder than it actually is. It's a lot of steps, but most of them are just this acyl transfer and then the regeneration using our electron carriers. All right, then now we can go into the citric acid cycle. There are about 8, you can call it 10 steps of the citric acid cycle, where there are going to be different substrates, different products, different enzymes utilized, but then it's a cycle. It doesn't just create one thing at the end. The last step is the oxaloacetate, but the oxaloacetate can be reused over and over again. So we got into the citric acid cycle by taking that pyruvate, decarboxylating, and attaching on to a coenzyme A. And then eventually, we'll talk about that in the next lecture, it's going to be condensed with the oxaloacetate created at the end of the citric acid cycle to create citrate. So then that's where the citric acid cycle comes from. It's the condensation of acetyl-CoA, the acetyl group, to the oxaloacetate to create citrate. So we go from a 2-carbon thing, plus a 4-carbon thing to get our 6-carbon thing. And so it goes through a lot of changes where we get 2 carbon molecules, 3 carbon molecules, 4 carbon, 5 carbon, 6 carbon molecules. So not quite as straightforward as glycolysis, but since you've done glycolysis, it should be easier to catch on. So eventually the 2 carbons are oxidized to CO2, so we're going to lose a couple CO2s in the citric acid cycle. The 4 carbons that are left are ready to accept another acetyl-CoA, so it can go through that cycle. And given enough time, one molecule of oxaloacetate can oxidize any number of acetyl groups. So it's a cycle and it keeps being utilized over and over again. So for next time, make sure you're ready to learn all the enzymes and substrates and molecules involved in this cycle. We learned how to get into the cycle, but once we get in there, it's going to be reaction after reaction that are going to hopefully and prayerfully come easy to you. All right, that's it for today. I hope you stay safe and stay healthy.